All right, cell division. So we're going to look at the different ways that cells can divide. We'll start by talking about the prokaryotes, which, remember, are the cells that don't have a nucleus. They're essentially what we call bacteria cells, and they're a little bit simpler, so they have a simpler method for dividing. So if we take a bacteria cell and we think of it as a little membrane with like an envelope, and inside is the genetic material, right? The genes, we'll call that the chromatin. So chromatin refers to the genetic material. It's the molecules that contain the instructions, or the genes. And that essentially is, DNA. That's what we call deoxyribonucleic acid. Okay, so let's take a little look at this DNA molecule. We'll draw a little picture. It's got a little backbone like this, and then it has 4 different little molecules that can line up here. And they come in patterns. I'm just drawing an arbitrary pattern. There's no special pattern here. Arbitrary pattern. But there are 4 different types. That's the catch. So we'll draw a green one here. It doesn't really matter. Maybe another blue one over here like this. And of course, these little molecules line up on the DNA, and they spell out the instructions of our genes. So essentially, each little group of 3 forms a little genetic word. And then you can put many groups of 3 together to form genetic sentences that spell or say things, just like you would use letters to spell words and then put words together to write out thoughts or ideas. So it's the pattern of molecules on this molecule, this DNA, the pattern of these little molecules inside of it that spell out our genes. Now there's a second section. I've drawn another line here because this molecule has another side to it. And on the other side, there will be the same molecules, but they always match up in a certain way. Blue and red will always be opposite each other, like this. And then purple and green will always be opposite each other, like this. And that's because the shape of these molecules is such that they fit together very nicely, and they form a little weak bond when they're together. So this is what DNA kind of looks like. It has patterns of molecules along a backbone. The backbone is made of other molecules. It's made of something called ribose, a kind of ribose, sugar, it's actually a kind of sugar, and phosphates, which we've learned about in chemistry. So it's a complicated molecule, but this is sort of a nice little picture that gives us an idea of how it will replicate itself. DNA has the ability to replicate a copy of itself. So what you have to do is you have to imagine this DNA is floating in the nucleus. And of course, the nucleus has fluid inside, liquid. It's like a soup. And in that soup, there are all kinds of molecules. So all these little molecules are floating around. The blue ones are floating around. The red ones are floating around. The purple ones all over the place. And the green ones. But they're not really doing anything as of yet because they're not part of any molecule. It's like your Lego, right? You're building your spaceship out of Lego. You still have little bits of Lego lying around ready to be used. So what happens is this molecule, which normally has a very, very sort of light attraction between here, it can basically unzip itself, can open itself up. So when it unzips itself, here's what it does. Let me go a little more space here. It unzips itself so that the top breaks away from the bottom. Okay, let me get my line back. Get that, make it a little thicker. There's the same thing, but it's going to unzip. And then down here, it's going to unzip itself like this. So it's opening up. So now I'll just draw what I had before up on top. I had a blue one there and a green one here. And then I have these 2 purple ones. And then a red one. Remember, these are just molecules in order. And then a blue one. And then another red one. I'll just extend that a bit. another red one. But it's broken apart. So down below, I have a blue one here that used to be attached to that red one. There's another blue one over here. And then there's a red in the middle. So they've pulled apart. And then over here, I've got the green one and the green one. So remember, you have to make sure they match up. And then there's a purple. This one might still be attached because it hasn't opened up yet, hasn't unzipped. And then what happens is, also floating around in here are little bits of molecules like the ribose and the backbone molecules that I talked about. They're all floating in the soup, right? The cytosol soup. And so these little guys, now that it's opened up, these little guys can float into place. And this green one here will just float in here, and it'll attach to where the other purple one was. And this red one will float in, and it'll attach to this blue. And on the bottom, this purple one will float in and attach to the green. So what happens is you get this kind of thing happening. I'll erase those marks. You basically get little purple molecules lining up right here and there, like that. And then the red one would come along and line up over here, and another one here, and a blue one sort of in the middle there, and something like that. And then along the back, little bits of the backbone would start to form, and it would go like this. And then on the top, the same thing would begin to happen. Okay? So on the top, you'd get a green attaching here, and a green attaching here, and then a blue here, and a blue here, and a red over here, and little bits. Oh, I guess I need little sticks on them. Little sticks. And these, of course, would be attaching to a new strand like this. So you can see what happens if you look at the two pieces that we've created, right? If you look at this entire piece right here and this entire piece right here, and you compare them to the original piece, you see that they're exactly the same. They have the exact same pattern on the top and the bottom. And so what we've effectively done is by opening up this molecule and rebuilding the bottom half and the top half, we create two copies of the same molecule that we had when we started. So any instructions or patterns that were in the first one are maintained in the second one. And this continues down the molecule until the genes have copied themselves. And so that's an elaborate process that you learn about more in your grade 11 and 12 classes. But for us, that's just enough to get us to understand the basic idea of how your genes can copy. So now the genes can copy, and then once you copy the genes, the chromatin, then all you have to do is basically split apart. So for the bacteria, the process is actually very simple. The process is called binary fission. Binary means two, and fission means to split. So this simply means to split in two. And so what happens is we have genetic material inside. The first thing that would happen is the genetic material would copy itself in the process that we just showed. And so now you have copies, right? DNA copies itself, or the word we use is replicates. That's the word we like to use. Replicates. DNA replicates. And then the cytoplasm begins to stretch. The membrane, remember the membrane of the cell is very fluid, and so it can stretch and twist, and it pinches off. So the next step would be sort of like, it would start to go like this. And one copy of the genes would go one way, and the other copy would go the other way. And then you can see how there's this pinching action here. And eventually when they touch, they would just pinch off, and you produce two bacteria cells with the same genetic information inside. And that's basically the process of binary fission. You duplicate the DNA, and then you duplicate the actual cell by splitting it in two. Now, there's a little more to the process. We're just looking at the beginnings here, the basics, so we can differentiate. When you get into eukaryotic cells, though, it's a little more complicated. Because in eukaryotic cells, you've got a nucleus. The DNA is stuck in the nucleus. There's all those organelles. It's a little more complex. So let's talk about what happens in eukaryotic cells next. Okay, eukaryotic cells, eukaryotes, more complicated. They also divide, but it's similar to the process of binary fission. So you can see how in simpler cells like bacteria, it could evolve into a more complicated process because it shares a lot of the same elements. We call the process mitosis. That's the process of division for eukaryotic cells. Division process. That's its name. So to understand how it works, we first have to understand sort of the life cycle of a cell. So before we can talk about how it divides, let's just draw a little picture here, and we'll call it the cell cycle. And so what happens is cells, they go through a process where they divide and split into two. So we'll put a little black line here across the top like this, and we'll write mitosis on here. So there's the division stage of a cell. And then once the cells have divided, they're like little baby cells, they begin to go through a phase of growing. And so we call it the G1 phase, G1. It actually stands for gap phase. But if you think about growth, gap, and G, they all start with the same letter. It gives you a hint. Then what happens is there's when the cells have now grown, where they're nearly full grown, they're doing their jobs, whatever their jobs might be. If they're a liver cell, they're breaking down toxins or doing all kinds of things, whatever they do inside the body. Or if they're a single cell, all the jobs of life being performed. And then they go into a phase where they begin to replicate. This is called the S phase, which stands for synthesis. Synthesis. This is the phase where the chromosomes do their thing and double up. So it's the chromosome replicating phase. Okay? I should say the DNA replicating phase, because I haven't explained what chromosomes are just yet, but I will soon. So that's when the DNA copies itself. And then we have another growth phase after that, the G2 gap phase, where the cell continues to grow and then gets ready to start all over again and split into and divide and carry on. So the black part is the dividing part, and the green parts are the growing phases and doing their job phase, and then the blue synthesis phase is the point where the DNA copies itself. So you notice how the DNA kind of copies itself, not at the end of the cell's life, but sort of before the cell is ready to divide. And there's another little growing phase after that. So sometimes the cell has a single copy for most of its life, but then there's a period after where it has a double copy as it prepares to get ready to divide. So the cell cycle, okay? So maybe we should just write a few things there just to describe what we said. This is a growing phase. And also growing phase, but not just growing, but doing cell jobs. That's what the cell does. It's basically doing its work throughout that phase. And then on the other side, we also have another growing phase where the cell continues to grow to its full size, but it's also preparing to divide during this phase, towards the end. And then in the blue phase, the synthesis phase, which I'm going to write that under here, synthesis. Synthesis means to form or to make. And so what's happening is we are forming or making copies. DNA copies itself in this phase. And that's important. So it actually gets a little confusing when we're talking about cells, trying to keep track of whether they have a single copy or a double copy of their genes at any given time. So it's a little confusing. All right. Now we're going to look at that black phase. at mitosis. We're going to cut it up into pieces. So first of all, I'm going to show you the main stages. We take that black arrow, I blow it up like this. What we find is it comes in stages. And the first stage is called prophase. So mitosis has stage 1, prophase. The second stage is called metaphase. The third stage is called anaphase. And the last stage is called telophase. Telophase. Some people say telophase. Telophase or telophase. So that black little division process that we had up here at the top, where it says mitosis, that's what this is. So I'll write mitosis right across the top, because mitosis has these 4 stages in it. And we also break down the stages into sub-stages, like we'll say, early prophase or pro-metaphase, but we're not going to worry about that just yet. We're going to look at the 4 main stages and what's going on and see what happens. Okay, so those are the phases. Let's see what happens then and how it works. So, first of all, in order to understand cell division, we have to also understand another structure of the cell. So you'll remember, I'm going to write here microtubules. You'll remember we talked about cells, and we said that inside the cell, there's all these structures, right? There's the nucleus, and there's the endoplasmic reticulum, and there's little sausage-shaped mitochondria, and all these things. There's another thing inside the cell. It's called the cytoskeleton. Microtubules form a part of the cytoskeleton. Now, they're not made of bones, the skeleton. They're actually made of proteins, protein molecules. But they are rigid protein molecules. They're tube-shaped. And what they do is they give some structure to the cell, so it's not all floppy. They can actually make the cell have some shape, help it hold its shape. And so these microtubules are associated with a couple of structures that are inside the cells. So the cells have what's called a centrosome, which is actually made of 2 centrioles. So I think on the picture that we drew, we called it the centrioles. We drew the 2 little things sort of stacked together like an X, remember? And they had lines on them because they're actually tube-like structures. So a centrosome is a little thing that has 2 centrioles in it. And we said that they were going to play a role in the division of cells. So these organelles, their job is to control the cytoskeleton. And they can actually initiate the growth of these tubules, which can stretch out through the cell and go in different ways. And they have many purposes. They keep the cell's shape. In some cells, they actually help it move around. But in the case of division, they have a special role to play. They form these microtubules, and we're going to see that they have an important role. So just remember that when we get to that point in the story. Okay, so stage 1, prophase. In this case, we have a cell, and inside the cell, we have the nucleus. Now, the nucleus contains the DNA. So there's a couple things that are going to start to happen. I'm going to draw the nucleus sort of large and with a dotted line like this. I'm drawing it large so we can look inside and see what's happening, okay? Inside the nucleus, there is DNA. Now, the DNA in eukaryotic cells is usually wrapped up in something called a chromosome. DNA is packaged in a chromosome. So what the DNA molecule is it's actually a long, windy molecule. It's very long, many meters long. It's hard to believe, though, you can have a molecule that's meters long and it's inside the nucleus of a microscopic cell. And so typically the DNA is a very thin, long strand like this. And it's wrapped up in proteins. So the DNA is packaged in the chromosome with protein molecules. They're called histones. And they just basically regulate the shape of the molecule. Now, to save space, they're all stretched out. But when it comes time to divide, something really interesting happens. The DNA molecule gets really short, and it gets all sort of pulled in, and it sort of turns into a long sort of sausage-shaped thing like this. And then, of course, when it copies itself, it makes a second copy, and the second copy is sort of attached to the first copy, something like this. So when we draw that, that's two copies. It's two identical chromosomes. And they're attached at this point right here, which is something called a centromere. So let's just, let's write centromere. So this, it gets a little confusing, right? Because you have to remember, is it one copy or two? So a chromosome is typically one copy. So this is a chromosome that we've drawn here. But at cell division time, the chromosome has already been doubled, because that happened way back in the S phase, remember? All right, so we have a single chromosome, but then it doubles and it looks like this. So we still call it a chromosome, even though it's got two copies, but we give the names to the parts. So the copied chromosome, each part is called a chromatid. Let me just rewrite that. A chromatid. So that's one chromatid on this side, and this is one chromatid on the other. Okay? Chromatids are just, we call them sister chromosomes. They're exactly the same. In fact, what we do in the lab is when we add stains to cells to make things show up better, we find that when you stain these chromosomes, they stain in the interesting patterns of like little bands like this. And there'll be dark spots and light spots and dark spots and light spots, and they all have a certain pattern. But of course, because they're identical copies of each other, the other one has exactly the same pattern. And they look exactly the same, or at least very much the same, as we'll see a little bit later what can happen here. Okay, so the chromosome is now in a doubled form. So this is the single form of the chromosome. But then we have the double form. So let me put this over here, double form, double copy. And this is usually how we see them in pictures, because that's the only way they're ever big enough for us to see. When they're all stretched out in their single form and they're very thin, right? Now they can double in the thin form as well. So we could add another one to show it doubling. But it's when they get really thick like this that we can actually see them in the microscope. Otherwise you can't see them. They're too thin and too stretched out. And so this is how we usually see chromosomes. We always see them in their double copied form, which is why they look like that. The little thing that joins them is called the centromere. The centromere is what right there that joins them together, holds on to them. So they're stuck. The two copies aren't just let to go for free. You don't just copy yourself and then fall apart. They remain stuck together, and that's very important in the process. So let's draw ourselves a couple of chromosomes inside here, and I'm going to use some different colors. Okay. So here's a little blue one, and there are the two chromatids, and it's stuck together at the centromere. Here's a little purple one, and we'll draw a little red one. Okay, maybe I'll make it green. I'll make it green so it stands out from the membrane. Okay. So, in prophase, here's what's happening. The two important things that are happening. Number one, the chromosomes are going from their sort of long, skinny, stretched out form to their short, thick, fat form. They're shortening up and they're thickening. So they're turning into these guys down here. And they become visible in the microscope where we can actually see them. The other thing that's happening is the membrane around the nucleus is beginning to fall apart. That's why I drew it in a dotted line. the red dotted line is the nuclear membrane, but it's beginning to dissolve or fall apart, and so therefore it won't hold the nucleus any longer. Okay, so in prophase, we have two things happening. We have the shortening and thickening of chromosomes, and we have the breaking down of the nuclear membrane. There's actually something else I should point out here too. So there's actually more than two things. Remember those centrosomes we talked about? Well, the centrosomes, they copy themselves also. So where you had one centrosome, it copies itself and makes two. And so one centrosome begins to work its way around one copy, goes this way towards the top of the cell, and the other centrosome works its way down here. So let's write here that the centrosomes copy, centrosome, the centrosome, whoops, gotta spell it right, the centrosome replicates, let's use that word, it's a nice big word, replicates, and each one moves to what we call the poles of the cell, just like the north and south pole of Earth. We call the top and the bottom the north and south pole. We just call them the poles of the cell. And while they do this, they begin to grow or to form what are called spindle fibers. These are microtubules that begin to stick out and grow out from the centrioles. So we'll add to our picture. Let me draw the centrioles a little larger here. And what's happening is these tiny little tubes are starting to grow outwards like this from the centrioles, or the centrioles inside the centromeres, or sorry, the centrosomes, yeah. So we got to keep it straight. Centromere joins chromosomes together, or joins chromatids in the double copy form of a chromosome. The centrosome is a little body in the cell that has two centrioles inside. So when it copies, each one has two centrioles inside, and they form these little fibers called spindle fibers. So that's what's happening in our prophase. Okay. We have time to get to one more phase. Let's do metaphase next. And then we'll have to break this video and we'll do a second video to do the rest. So we have metaphase next. All right. Here's what's really cool. In metaphase, here's my cell. The nuclear membrane is gone. It dissolved. So we have our three chromosomes. I'll keep the same colors. So we had what? We had a purple, a blue, and a green chromosome. But now, the spindle fibers that were reaching out from the centrosomes are going to attach to the chromosomes. So we had, and the chromosomes themselves are being pushed and guided by these little tubules, these spindle fibers, to go to the middle of the cell. And they line up here in the middle like this. And then you have the centrosomes on either end of the cell. And what you have are these little spindle fibers that have grown all the way out and are attaching like this to this side. And then they're also growing out and attaching to this side like this. So what's happening here is the chromosomes are lining up in the middle of the cell. Let's just say in the middle. And the reason they're doing that is they're being guided that way by these little spindle fibers, these microtubules that are pushing them into place, the middle of the cell. And secondly, the other thing that's happening is the spindle fibers are now fully formed. Fully formed and attach to each chromatid. You can see in the picture. Okay, actually maybe we can get to anaphase. Maybe we can get finished if we have time here. So the next stage would be the anaphase. Anaphase. So what happens here is the cell begins to stretch. Whoops, let me go back there. I'm going to need a little more space here because we have to draw this sort of stretched out. So the cell begins to stretch itself out in an elongated way, like this. The cytoplasm begins to push, the microtubules stretch the cell. And what happens is, here's my little centrioles in here, is as it stretches out, the microtubules pull on the chromatids and they come apart. So I got to get my colors right, blue, red, purple. So the little blue chromatid gets pulled away from the other like this. And same with the red. And they're pulled from the middle, so they form these little V-like shapes. Here are the, whoops, that's supposed to be black. These are the spindle fibers that are pulling and stretching, so they've come apart. You can see how they've broken apart, which means the copies are now being pulled to two different parts of the cell. So here we have the spindle fiber, well, let's put the cell elongates, and the spindle fibers pull the chromatids, right, which are essentially the DNA copies of, that's what they are, apart, pulls them apart, like this. And then we have one more stage, telophase. And in telophase, you can kind of guess what's happening here, right? We've separated the copies of the genes, now we just have to separate the rest of the cell. So in telophase, we see this beginning to happen. The cell starts to pinch in on itself, the elongated cell. And the nuclear membrane starts to come back, starts to form around both. And now in each one, we have a little wiggly blue chromosome, we have a little wiggly purple one and a little wiggly green one that were pulled in there. A little green one, a little blue one down here, and a little purple one here. So what's happening now is the nuclear membrane begins to reform, reforms, right? But it reforms in two different places because we're forming two different cells. And then the cytoplasm separates like this. We call this cytokinesis. Cytokinesis. The movement of the cytoplasm. Kinesis means to move. So what's happening is these are pinching inwards. And eventually what will happen is this will lead to a complete pinching off at the end of telophase. So this would be the early telophase. But what this turns into, of course, is one full cell here, one full cell here that have separated. They each have a fully formed nucleus. And inside that fully formed nucleus, they each have a copy of the original chromosomes ready to go. So we call these the daughter cells. These are the daughter cells, and there are two daughter cells produced here by this process. You'll notice though that they're identical to the original cell that we started with, if we go back to the beginning. And that's essentially the 4 stages of mitosis. And now these two cells will begin their G1 phase, and they will grow and do their job and eventually get to the point where they can replicate themselves again. So we'll stop there, and we'll call that a day.